Antisense RNA strategies for regulating gene expression. Antisense RNA is RNA, also referred to as antisense transcript, natural antisense transcript, or antisense oligonucleotide B, is a single-stranded RNA that is complementary to a protein coding messenger RNA with which it hybridizes and thereby blocks its translation into protein. The AsRNAs, which occur naturally, have been found in both prokaryotes and eukaryotes and can be classified into short and long non-coding RNAs. The primary function of AsRNA is regulating gene expression. AsRNAs may also be produced synthetically and have found widespread use as research tools for gene knockdown. They may also have therapeutic applications. Antisense RNA molecule represent a unique type of RNA transcript that comprises 19 to 23 nucleotides and is complementary to mRNA. Antisense RNAs play a crucial role in regulating gene expression in multiple levels, such as replication, transcription, and translation. Antisense RNA technology is one of the approaches that are used for the inhibition and gene expression of downregulation of a gene. This technology works on the principle that an antisense nucleic acid sequence base pairs with its complementary sense RNA strand and prevents it from being translated into protein. The complementary nucleic acid sequence can be either a synthetic oligonucleotide, often oligodeoxyribonucleotides of less than 30 nucleotides, or longer antisense RNA sequences. The complementary sense and antisense RNA molecules in the same cell can lead to the formation of a stable duplex, which may interfere with gene expression at the level of transcription, RNA processing, or possibly translation. Antisense refers to the orientation of complementary strands during transcription. The two complementary strands of DNA are referred to as sense and antisense, or coding and non-coding. Transcription uses the antisense strand as template, resulting in an mRNA that is identical in sequence to the sense strand, except for the replacement of uracil for thymine. Antisense RNA is synthesized using the sense strand as template, therefore it is a sequence complementary to mRNA. Sense strand, tRNA, transcribe antisense, transcribe sense. Normal mRNA, antisense mRNA. Transcribe sense then means antisense mRNA. Normal and antisense RNA can anneal. Antisense RNA sequence are complementary to a target mRNA. Antisense RNA forms double-stranded regions that block either protein translation or splicing of introns. Antisense RNA technology is a method used to silence genes and inhibit their expression. Gene function relies on expression, which follows the central dogma of molecular biology. The use of AsRNA can regulate gene expression by targeting specific mRNAs, which can result in changes in phenotype, disease resistance, and other traits associated with the protein expression profiles. This technology uses short, single-stranded oligonucleotide strands that are complementary to the targeted mRNA. Antisense technology works on the principle of complementary base pairing between antisense oligonucleotide sequence and its corresponding sense RNA strand. This binding impedes translation, the ultimate step in protein synthesis, by blocking the sense RNA strand from participating in the process. The messenger RNA is responsible for transmitting genetic information from the DNA to other cellular components involved in protein production. To put it simply, antisense technology uses complementary DNA base pairing to halt protein production by interfering with mRNA function. The antisense approach aims to decrease the expression of a specific molecular target. This is accomplished by triggering the activity of RNase H endonuclease, which cleaves the RNA duplex formed between the antisense DNA and the mRNA of the target gene in the cytoplasm. This cleavage leads to a reduction in protein synthesis and significant decrease in the translation of the target gene. The primary strategy for ASON's gapmers is to trigger RNA H activation while which cleaves the mRNA's RNA portion and the antisense RNA duplex, resulting in the degradation of the target mRNA. Furthermore, ASONs that do not cause RNA H cleavage can impede translation by physically obstructing the ribosome, known as steric blockers. When targeting a 5' terminus with ASONs, it is possible to prevent the binding and assembly of the translation machinery. Then antisense RNA regulation strategies. Antisense RNA strategies are a set of techniques used to regulate gene expression by targeting specific RNA molecules. The strategies involve the use of antisense oligonucleotides or antisense RNA molecules to interfere with the expression of target genes. Antisense oligonucleotides, ASOs, are short single-stranded DNA or RNA molecules that are designed to be complementary to a specific target RNA sequence. By binding to the target RNA, ASOs can prevent prevent translation or promote degradation of the RNA molecule, thereby reducing the expression of the corresponding gene. ASOs can be delivered into cells using various methods such as transfection or viral vectors. Small interfering RNA, siRNAs, are double-stranded RNA molecules typically generated from longer hairpin precursor molecules. Once outside the cell, siRNAs are processed into small RNA duplexes that guide the RNA-produced silencing complex to the target mRNA. The RISC complex then cleaves the target mRNA or inhibits its translation, leading to reduced gene expression. Then short hairpin RNAs or shRNAs. shRNAs are designed to mimic the structure of naturally occurring microRNAs or miRNA. They are usually expressed from DNA plasmids or viral vectors and proceeds in the gel to generate small hairpin RNA molecules. These hairpins are then cleaved into small interfering RNA that mediate gene silencing through the RISC complex, similar to siRNAs. Then antisense mediated exon skipping. This strategy is commonly used to modulate splicing of pre-mRNA molecules by targeting specific splice sites or exonic sequences, not exotic, exonic sequences. Antisense oligonucleotides can disrupt the normal splicing process, leading to the skipping of specific exons during mRNA maturation. Exon skipping can be used to correct genetic mutations or modulate gene expression by removing specific exons from the mature mRNA. Antisense RNA-based gene activation. In addition to gene expression, antisense RNA molecules can also be used to activate gene expression. By designing antisense molecules that target non-coding regulatory elements or proper regions, it is possible to modulate gene transcription and enhance gene expression. These antisense RNA strategies often offer powerful tools for studying gene function, modulating gene expression levels, and developing potential therapeutic approaches for various genetic disorders. However, it is important to consider factors such as delivery methods, stability, and specificity when designing and implementing antisense RNA-based strategies. Then siRNA and miRNA regulation. The downregulation of gene expression by miRNA and siRNA is a complex process involved both translational repression and accelerated mRNA turnover, each of which appears to occur by multiple mechanisms. Moreover, under certain conditions, miRNA are also capable of activating translation. A variety of cellular proteins have been implicated in this regulatory mechanisms, yet their exact roles remain largely unresolved. Then siRNA. Small interfering RNA or siRNA, sometimes known as short interfering RNA or silencing RNA, is a class of double-stranded RNA at first non-coding RNA molecules, typically 20 to 24, normally 21 base pairs in length, similar to miRNA and operating with the RNA interference pathway. It interferes with the expression of specific genes with complementary nucleotide sequences by degrading mRNA after transcription, preventing translation. Schematic representation of siRNA molecule. The naturally occurring siRNA have a well-defined structure, that is a short, usually 20 to 24 base pair double-stranded RNA, dsRNA with phosphorylated 5' ends and hydroxylated 3' ends with two overhanging nucleotides. The dicer enzyme catalyzes production of siRNA from long dsRNAs and small hairpin RNA. siRNA can also be introduced into cells by transfection, since in principle the gene can be knocked down by a synthetic siRNA with a complementary sequence. siRNA are an important tool for validating gene function and drug targeting in the post-genomic era. The siRNA silencing genes by inducing degradation of the target mRNAs, while the miRNA tend to silence genes by interfering with accumulation of the protein products of the target mRNA. However, if base pairing between the animal miRNA and its target mRNA is perfect or near perfect, the miRNA causes cleavage of the target mRNA. The siRNA are formed by dicer action on double-stranded RNAs that usually contain at least one strand that is foreign to the cell, derived from transposons. On the other hand, the miRNA is formed by dicer action on the double-stranded part of stem loop RNA, that is a normal cellular product. The siRNA base pair perfectly with the target mRNA, whereas the miRNA usually base pair imperfectly with their target mRNAs. The mechanism of siRNA action, the method by which siRNA causes the silencing of genes is as follows. A double-stranded RNA is cleaved by the dicer enzymes. This forms siRNA. The double-stranded siRNA enters the cell and forms the RNA-reduced silencing complex or RISC with other proteins. This is unwound, which forms the single-stranded siRNA. The strand of RNA with the 5' end base pairing that are thermodynamically less stable remains part of RISC complex. This strand can now scan for complementary mRNA. Once this antisense strand binds to the target mRNA, mRNA cleavage is induced. The foreign mRNA is recognized by the host cell as abnormal and is degraded. Now translation is possible, and therefore the gene is silenced. siRNA have very high target specificity as they cleave, and mRNA before translation compared to the similar miRNA which silences genes by repressing translation. siRNA has 100% complementarity to its target mRNA. siRNA has also been found to activate gene expression. The mechanism by which natural siRNA causes gene silencing through repression of translation occurs as follows. Long dsRNA, which can come from hairpin complementary RNAs, and DNA-dependent RNA polymerase is cleaved by an endoribonuclease called dicer. Dicer cuts the long dsRNA to form short interfering RNA or siRNA. This is what enables the molecules to form the RNA-induced silencing complex. Once siRNA enters the cell, it gets incorporated into other proteins to form the RISC. Once siRNA is a part of the RISC complex, the siRNA is unwound from the single-stranded siRNA. The strand that is thermodynamically less stable due to its base pairing at the 5' and is chosen to remain part of the RISC complex. The single-stranded siRNA, which is a part of RISC complex, now can scan and find a complementary mRNA. Once the single-stranded siRNA part of the RISC complex binds to its target mRNA, it induces mRNA cleavage. The mRNA is now cut and recognized as abnormal by the cell. This causes degradation of the mRNA and in turn no translation of the mRNA into amino acid and then proteins, thus silencing the gene that encodes that mRNA. siRNA is also similar to miRNA. However, miRNAs are derived from shorter stem loop RNA products. miRNA typically silence genes by repression of translation and have broader specificity of action, while siRNAs typically work with higher specificity by cleaving the mRNA before translation with 100% complementarity. So dsRNA and hairpin dicer siRNA duplex formation of RISC siRNA mRNA complex silenced mRNA gene silencing will be taking place. Then about miRNA, another class of small RNA called the microRNAs. miRNAs are 22 RNA produced nucleotide or RNA produced naturally in plant and animal cells by cleavage from a large stem loop precursor. In animals, this miRNA then base pair, though imperfectly, with 39 untranslated regions of specific mRNAs and silence gene expression primarily by blocking translation of those mRNAs. miRNA or microRNA are short non-coding RNAs that regulate gene expression post-transcriptionally. They generally bind to the 3' UTR untranslated region of their target mRNAs and repress protein production by destabilizing the mRNA and translational silencing. MicroRNA is the name of the family of molecule that helps cells control the kinds and amounts of proteins they make. That is, cells use microRNA to help control gene expression. So microRNAs or miRNAs is a subset of non-coding RNAs are 22 long endogenously initiated short RNA molecules that are considered to post-transcriptionally regulate the cleavage of target mRNAs or just to repress that translation. miRNAs are thought to be important regulators of gene expression and act via dynamic mechanisms that are influenced by molecular localization and mechanisms of interaction with the targets. And mechanism of action of microRNA. MicroRNA bind to target mRNAs through sequence complementarity, leading to changes in the translation pattern of mRNA proteins. miRNA bind to their targets primarily at untranslated region, 3' UTR. Alternatively, miRNA bind to another region such as 5' UTR or even a promoter region in the target mRNA. Binding site determines the biological consequences of these interactions. Most miRNA inhibit gene expression by hindering target mRNA protein translation. Interactions between miRNA and target mRNA may lead to mRNA degradation through cleavage mediated by endonuclease in a RISC to which the miRNA is bound. In animal cells, most miRNAs are normally partially complementary to target mRNAs, which seem to be enough to permanently inhibit target mRNA translation. The small size of miRNA and tolerability of partial complementarity with its targets allows interactions with many mRNAs. The major difference between siRNAs and miRNAs is that former inhibit the expression of one specific target mRNA, while the latter regulate the expression of multiple mRNAs. siRNA and miRNA inhibit translation by two different mechanisms while working in association with the protein, forming a ribonucleoprotein complex called RNA-induced silencing complex. The protein in RISC unwind siRNA and remain bound to a single antisense strand which then bind to mRNA in a sequence-specific manner, at which time a protein component of RISC called slicer cuts the mRNA in the middle of the binding region. The cut mRNA is recognized by the cell as being abnormal and is subsequently destroyed, and in case of miRNA, the microRNA-induced silencing complex associated with the mature miRNA, and the complex binds to mRNA and physically blocks the translation. Many miRNA form perfectly complementary stem loop structures on the target sense strand of mRNA as opposed to siRNAs, which require near perfect matches. In general, only one miRNA is produced from one precursor. In contrast, siRNA is proposed to moderate its own amplification in plants and certain animal species such as C. elegans. Proposed model suggests that either the double-stranded mRNA siRNA hybrid or the sense strand of siRNA, which is released by RISC, undergo elongation or transcription respectively by RNA-dependent RNA polymerase to generate new double-stranded piece of RNA, which acts as a new substrate for dies and can ultimately lead to the formation of new RISC.